Hey everyone, welcome to NCRT Audiobook, Chapter 20 Locomotion and Movement. Movement is one of the significant features of living beings. Animals and plants exhibit a wide range of movements. A streaming of protoplasm in the unicellular organisms like amoeba is a simple form of movement. Movement of cilia, flagella, and tentacles are shown by many organisms. Human beings can move limbs, jaws, eyelids, tongue, etc. Some of the movements result in a change of place or location. Such voluntary movements are called locomotion. Walking, running, climbing, flying, swimming are all some forms of locomotory movements. Locomotory structures need not be different from those affecting other types of movement. For example, in paramecium, cilia helps in the movement of food through cytopharynx and in locomotion as well. Hydra can use its tentacles for capturing its prey and also use them for locomotion. We use limbs for changes in body postures and locomotion as well. The above observations suggest that movements and locomotion cannot be studied separately. The two may be linked by stating that all locomotions are movement but all movements are not locomotions. Methods of locomotion performed by animals vary with their habitats and the demand of the situation. However, locomotion is generally for search of food, shelter, mate, suitable breeding grounds, favorable climatic conditions, or to escape from enemies, predators. 20.1 Types of movement. Cells of the human body exhibit three main types of movements, namely amoeboid, ciliary, and muscular. Some specialized cells in our body, like macrophages and leukocytes in blood, exhibit amoeboid movement. It is affected by pseudopodia formed by the stemming of protoplasm, as in amoeba. Cytoskeletal elements like microfilaments are also involved in amoeboid movement. Ciliary movement occurs in most of our internal tubular organs, which are lined by ciliated epithelium. The coordinated movements of cilia in the trachea help us in removing dust particles and some of the foreign substances inhaled along with the atmospheric air. Passage of ova through the female reproductive tract is also facilitated by the ciliary movement. Movement of our limbs, jaws, tongues, etc. require muscular movement. The contractile property of muscles are effectively used for locomotion and other movements by human beings and majority of multicellular organisms. Locomotion requires a perfect coordinated activity of muscular, skeletal, and neural systems. In this chapter, you will learn about the types of muscles, their structure, mechanism of their contraction, and important aspects of the skeletal system. 20.2 Muscle. You have studied in chapter 8 that the cilia and flagella are the outgrowths of the cell membrane. Flagellar movement helps in the swimming of spermatozoa, maintenance of water current in the canal system of sponges, and in locomotion of protozoans like euglena. Muscle is a specialized tissue of mesodermal origin. About 40 to 50% of the body weight of a human adult is contributed by muscles. They have special properties like excitability, contractility, extensibility, and elasticity. Muscles have been classified using different criteria, namely location, appearance, and nature of regulation of their activities. Based on their location, three types of muscles are identified: skeletal, visceral, and cardiac. Skeletal muscles are closely associated with the skeletal components of the body. They have a striped appearance under the microscope and hence are called striated muscles. As their activities are under the voluntary control of the nervous system, they are known as voluntary muscles too. They are primarily involved in locomotory actions and changes of body postures. Visceral muscles are located in the inner walls of hollow visceral organs of the body, like the alimentary canal, reproductive tract, etc. They do not exhibit any striation and are smooth in appearance. Hence, they are called smooth muscles, non-striated muscle. Their activities are not under the voluntary control of the nervous system and are therefore known as involuntary muscles. They assist, for example, in the transportation of food through the digestive tract and gametes through the genital tract. As the name suggests, cardiac muscles are the muscles of heart. Many cardiac muscle cells assemble in branching pattern to form a cardiac muscle. Based on appearance, cardiac muscles are striated. They are involuntary in nature as the nervous system does not control their activities directly. Let us examine a skeletal muscle in detail to understand the structure and mechanism of contraction. Each organized skeletal muscle in our body is made of a number of muscle bundles or fascicles held together by a common collagenous connective tissue layer called fascia. Each muscle bundle contains a number of muscle fibers. Each muscle fiber is lined by the plasma membrane called sarcolemma enclosing the sarcoplasm. Muscle fiber is a syncytium as the sarcoplasm contains many nuclei. The endoplasmic reticulum, that is, sarcoplasmic reticulum of the muscle fibers, is the storehouse of calcium ions. A characteristic feature of the muscle fiber is the presence of a large number of parallelly arranged filaments in the sarcoplasm called myofilaments or myofibrils. Each myofibril has alternate dark and light bands on it. A detailed study of the myofibril has established that the striated appearance is due to the distribution pattern of two important proteins, actin and myosin. The light bands contain actin and is called I band or isotropic band, whereas the dark band called A or anisotropic band contains myosin. Both the proteins are arranged as rod-like structures parallel to each other and also to the longitudinal axis of the myofibrils. Actin filaments are thinner as compared to the myosin filaments, hence are commonly called thin and thick filaments respectively. In the center of each I band is an elastic fiber called Z line which bisects it. The thin filaments are firmly attached to the Z line. The thick filaments in the A band are also held together in the middle of this band by a thin fibrous membrane called M line. The A and I bands are arranged alternatively throughout the length of the myofibrils. The portion of the myofibril between two successive Z lines is considered as the functional unit of contraction and is called a sarcomere. In a resting state, the edges of thin filaments on either side of the thick filaments partially overlap the free ends of the thick filaments, leaving the central part of the thick filaments. This central part of thick filament not overlapped by thin filaments is called the H zone. 20.2.1 Structure of contractile proteins. Each actin thin filament is made of two F filament actins helically wound to each other. Each F-actin is a polymer of monomeric G-actins. Two filaments of another protein, tropomyosin, also run close to the F-actins throughout its length. A complex protein, troponin, is distributed at regular intervals on the tropomyosin. In the resting state, a subunit of troponin marks the active binding sites for myosin on the actin filaments. Each myosin thick filament is also a polymerized protein. Many monomeric proteins called meromycin constitute one thick filament. Each meromycin has two important parts, a globular head with a short arm and a tail, the former being called the heavy meromycin and the latter the light meromycin. The HMM component, that is the head and short arm, projects outwards at regular distance and angle from each other through the surface of a polymerized myosin filament and is known as cross arm. The globular head is an active ATPase enzyme and has binding sites for ATP and active sites for actin. 20.2.2 Mechanism of muscle contraction. Mechanism of muscle contraction is best explained by the sliding filament theory, which states that contraction of a muscle fiber takes place by the sliding of the thin filaments over the thick filaments. Muscle contraction is initiated by a signal sent by the central nervous system via a motor neuron. A motor neuron along with the muscle fibers connected to it constitute a motor unit. The junction between a motor neuron and the sarcolemma of the muscle fiber is called the neuromuscular junction or motor end plate. A neural signal reaching this junction releases a neurotransmitter acetylcholine, which generates an action potential in the sarcolemma. This spreads through the muscle fiber and causes the release of calcium ions into the sarcoplasm. Increase in Ca2 plus level leads to the binding of calcium with a subunit of troponin on actin filaments and thereby remove the masking of active sites for myosin. Utilizing the energy from ATP hydrolysis, the myosin head now binds to the exposed active sites on actin to form a cross bridge. This pulls the attached actin filaments towards the center of A band. The Z line attached to these actins are also pulled inwards, thereby causing a shortening of the sarcomere, that is contraction. It is clear from the above steps that during shortening of the muscle, that is contraction, the I bands get reduced, whereas the A bands retain the length. The myosin releasing the ADP and PI goes back to its relaxed state. A new ATP binds and the cross bridge is broken. The ATP is again hydrolyzed by the myosin head and the cycle of cross bridge formation and breakage is repeated, causing further sliding. The process continues till the calcium ions are pumped back to the sarcoplasmic cisternae, resulting in the masking of actin filaments. This causes the return of Z lines back to their original position, that is, relaxation. The reaction time of the fibers can vary in different muscles. Repeated activation of the muscles can lead to accumulation of lactic acid due to anaerobic breakdown of glycogen in them, causing fatigue. Muscle contains a red-colored oxygen-storing pigment called myoglobin. Myoglobin content is high in some of the muscles, which gives a reddish appearance. Such muscles are called the red fibers. These muscles also contain plenty of mitochondria, which can utilize the large amount of oxygen stored in them for ATP production. These muscles therefore can also be called aerobic muscles. On the other hand, some of the muscles possess very less quantity of myoglobin and therefore appear pale or whitish. These are the white fibers. Number of mitochondria are also few in them, but the amount of sarcoplasmic reticulum is high. They depend on anaerobic process for energy. 20.3 Skeletal system. A skeletal system consists of a framework of bones and a few cartilages. This system has a significant role in movement shown by the body. Imagine chewing food without jaw bones and walking around without the limb bones. Bone and cartilage are specialized connective tissues. The former has a very hard matrix due to calcium salts in it, and the latter has slightly pliable matrix due to chondroitin salts. In human beings, this system is made up of 206 bones and a few cartilages. It is grouped into two principal divisions, the axial and the appendicular skeleton. Axial skeleton comprises 80 bones distributed along the main axis of the body. The skull, vertebral column, sternum, and ribs constitute the axial skeleton. The skull is composed of two sets of bones, cranial and facial, that totals to 22 bones. Cranial bones are 8 in number. They form the hard protective outer covering, cranium, for the brain. The facial region is made up of 14 skeletal elements which form the front part of the skull. A single U-shaped bone called hyoid is present at the base of the buccal cavity, and it is also included in the skull. Each middle ear contains three tiny bones, malleus, incus, and stapes, collectively called ear ossicles. The skull region articulates with the superior region of the vertebral column with the help of two occipital condyles, dicondylic skull. Our vertebral column is formed by 26 serially arranged units called vertebrae and is dorsally placed. It extends from the base of the skull and constitutes the main framework of the trunk. Each vertebra has a central hollow portion, neural canal, through which the spinal cord passes. First vertebra is the atlas and articulates with the occipital condyles. The vertebral column is differentiated into cervical 7, thoracic 12, lumbar 5, sacral 1 fused, and coccygeal 1 fused regions starting from the skull. The number of cervical vertebrae are 7 in almost all mammals, including human beings. The vertebral column protects the spinal cord, supports the head, and serves as the point of attachment for the ribs and musculature of the back. A sternum is a flat bone on the ventral midline of thorax. There are 12 pairs of ribs. Each rib is a thin flat bone connected dorsally to the vertebral column and ventrally to the sternum. It has two articulation surfaces on its dorsal end and is hence called bicephalic. First 7 pairs of ribs are called true ribs. Dorsally, they are attached to the thoracic vertebrae and ventrally connected to the sternum with the help of hyaline cartilage. The 8th, ninth, and 10th pairs of ribs do not articulate directly with the help with the sternum but join the 7th rib with the help of hyaline cartilage. These are called vertebrochondral false ribs. Last two pairs, 11th and 12th of the ribs, are not connected ventrally and are therefore called floating ribs. Thoracic vertebrae, ribs, and sternum together form the rib cage. The bones of the limbs along with their girdles constitute the appendicular skeleton. Each limb is made of 30 bones. Each limb is made of 30 bones. The bones of the hand, forelimb, are humerus, radius, and ulna, carpals. Wrist bones 8 in number, metacarpals, palm bones 5 in number, and phalanges, digits 14 in number. Femur, thigh bone, the longest bone, tibia and fibula, tarsals, ankle bones 7 in number, metatarsals 5 in number, and phalanges, digits 14 in number. are the bones of the legs, hind limb. A cup-shaped bone called patella cover the knee ventrally, kneecap. Pectoral and pelvic girdle bones help in the articulation of the upper and lower limbs respectively with the axial skeleton. Each girdle is formed of two halves. Each half of pectoral girdle consists of a clavicle and a scapula. A scapula is a large triangular flat bone situated in the dorsal part of the thorax between the second and the seventh ribs. The dorsal flat triangular body of a scapula has a slightly elevated ridge called the spine, which projects as a flat expanded process called the acromion. The clavicle articulates with this. Below the acromion is a depression called the glenoid cavity, which articulates with the head of the humerus to form the shoulder joint. Each clavicle is a long slender bone with two curvature. This bone is commonly called the collar bone. Pelvic girdle consists of two coxal bones. Each coxal bone is formed by the fusion of three bones, ilium, ischium, and pubis. At the point of fusion of above bones is a cavity called acetabulum to which the thigh bone articulates. The two halves of the pelvic girdle meet ventrally to form the pubic symphysis containing fibrous cartilage. 20.4 Joints. Joints are essential for all types of movements involving the bony parts of the body. Locomotory movements are no exception to this. Joints are points of contact between two bones or between bones and cartilages. Force generated by the muscles is used to carry out movement through joints where the joint acts as a fulcrum. The movability at these joints vary depending on different factors. Joints have been classified into three major structural forms, namely fibrous, cartilaginous, and synovial. Fibrous joints do not allow any movement. This type of joint is shown by the flat skull bones, which fuse end to end with the help of dense fibrous connective tissues in the form of sutures to form the cranium. In cartilaginous joints, the bones involved are joined together with the help of cartilages. The joint between the adjacent vertebra in the vertebral column is of this pattern and it permits limited movements. Synovial joints are characterized by the presence of a fluid-filled synovial cavity between the articulating surfaces of the two bones. Such an arrangement allows considerable movement. These joints help in locomotion and many other movements. Ball and socket joint between humerus and pectoral girdle, hind joint, knee joint, pivot joint between atlas and axis, gliding joint between the carpals, and saddle joint between carpal and metacarpal of thumb are some examples. 20.5 Disorders of muscular and skeletal system. Myasthenia gravis, autoimmune disorder affecting neuromuscular junction leading to fatigue, weakening, and paralysis of a skeletal muscle. Muscular dystrophy, progressive degeneration of a skeletal muscle mostly due to genetic disorder. Tetany, rapid spasm, mild contractions in muscle due to low calcium ions in body fluid. Arthritis, inflammation of joints. Osteoporosis, age-related disorder characterized by decreased bone mass and increased chances of fractures. Decreased levels of estrogen is a common cause. Gout, inflammation of joints due to accumulation of uric acid crystals. Summary. Movement is an essential feature of all living beings. Protoplasmic stemming, ciliary movement, movements of fins, limbs, wings, etc. are some forms exhibited by animals. A voluntary movement which causes the animal to change its place is called locomotion. Animals move generally in search of food, shelter, mate, breeding ground, better climate, or to protect themselves. The cells of the human body exhibit amoeboid, ciliary, and muscular movements. Locomotion and many other movements require coordinated muscular activities. Three types of muscles are present in our body. Skeletal muscles are attached to skeletal elements. They appear striated and are voluntary in nature. Visceral muscles present in the inner walls of visceral organs are non-striated and involuntary. Cardiac muscles are the muscles of the heart. They are striated, branched, and involuntary. Muscles possess excitability, contractility, extensibility, and elasticity. Muscle fiber is the anatomical unit of muscle. Each muscle fiber has many parallelly arranged myofibrils. Each myofibril contains many serially arranged units called sarcomere, which are the functional units. Each sarcomere has a central A band made of thick myosin filaments and two half I bands made of thin actin filaments on either side of it, marked by Z lines. Actin and myosin are polymerized proteins with contractility. The active sites for myosin on resting actin filament are masked by a protein troponin. Myosin head contains ATPase and has ATP binding sites and active sites for actin. A motor neuron carries signal to the muscle fiber which generates an action potential in it. This causes the release of calcium ions from sarcoplasmic reticulum. Calcium ions activate actin which binds to the myosin head to the form a cross bridge. These cross bridges pull the actin filaments, causing them to slide over the myosin filaments and thereby causing contraction. Calcium ions are then returned to the sarcoplasmic reticulum, which inactivate the actin. Cross bridges are broken and the muscles relax. Repeated stimulation of muscles lead to fatigue. Muscles are classified as red and white fibers based primarily on the amount of red colored myoglobin pigment in them. Bones and cartilages constitute our skeletal system. The skeletal system is divisible into axial and appendicular. Skull vertebral column, ribs, and sternum constitute the axial skeleton. Limb bones and girdles form the appendicular skeleton. Three types of joints are formed between two bones or between bone and cartilage: fibrous, cartilaginous, and synovial. Synovial joints allow considerable movements and therefore play a significant role in locomotion. That's all for today. Thanks for listening.